Welcome to the Triglyceride Paradox, understanding insulin resistance in Black Americans. So, a little bit of background first. The Triglyceride Paradox is a medical term used to describe a contradictory relationship between triglyceride levels and insulin resistance in Black Americans. Current screening tests for insulin resistance utilize the triglyceride HDL-C ratio. Triglyceride levels in the presence of insulin resistance in white Americans are elevated, while in Black Americans, triglyceride levels are low to normal. This is why we call it a triglyceride paradox. Triglyceride levels are an important risk factor for insulin resistance and diabetes. And it has already been established that there is a link between insulin resistance, diabetes, and the progression to cardiovascular disease. Black Americans are diagnosed with diabetes at a 60% higher rate than white Americans, which increases the risk of heart disease. Historically, medical data from non-white and diverse populations has been excluded from classic investigations examining metabolic syndrome and heart disease, namely the Framingham Heart Study, the Nurses' Health Study, and also the Physicians' Health Study. Health disparities among Black Americans are potentially made worse by their exclusion from clinical research. Why is this important? Well, so the CDC has analyzed some data that they got by assessing multiple racial and ethnic groups, including American Indian or Alaska Native, Black, Hispanic, White, and Asian or Pacific Islander. And so what they wanted is to investigate was how many deaths. They wanted to basically investigating the mortality rate associated with diabetes within each of these ethnic or racial groups. And they did it by assessing the deaths, the number of deaths per 100,000 people. And so as you can see here, Black Americans are second. They actually have the second highest mortality rate associated with diabetes, second to American Indian or Alaska Native. Studies demonstrate diabetes mortality is influenced by risk factors such as segregation, poverty conditions created by systemic racism, and lack of access to affordable health care. Let's go to our objectives. So, to determine which of the current screening tests will accurately predict the risks for insulin resistance in Black Americans. That's number one. The second is to identify other biological molecules other than triglycerides associated with insulin resistance. And additionally, we'd like to investigate the link between insulin resistance, diabetes, and cardiovascular disease in Black Americans. Questions and hypotheses. So two very important questions to ask before we pose the first hypothesis. So the first is, are more Black Americans underdiagnosed with insulin resistance due to the utilization of the triglyceride HDL-C ratio? So since paradoxically triglyceride levels are low to normal, and that will most certainly factor into the triglyceride HDL-C ratio. Black Americans who are assessed using this ratio, they may oftentimes come in below the threshold that medical professionals may use to diagnose individuals with insulin resistance. So it is highly likely. So the second one is there an alternative biomarker to measure broadly across diverse ethnic populations to assess or predict insulin resistance? There has been a lot of speculations about other biomarkers, but nothing has been produced definitively. So this leads us to our first hypothesis, which is if the triglyceride HDL-C ratio accurately assesses risks for developing insulin resistance in white Americans and fails to do so in Black Americans, and we evaluate other biomarkers of insulin resistance, then prevention of of type 2 diabetes will improve in both populations. Our next question. So, would increasing the efficacy in insulin resistance screening lower the presence of diabetes and the prevalence of diabetes and the risk of developing cardiovascular disease? This third question leads to our second hypothesis, which is if insulin resistance leads to diabetes and cardiovascular disease and screening tests are administered to insulin resistant patients as preventative measures, then fewer Black Americans will die from cardiovascular disease. Okay, let's move on to our aims. So our aim, number one, is to identify Black Americans in the US population living with insulin resistance. So we can probably do that by doing a meta-analysis. There are plenty of medical records, I believe, that can be made available through health databases. So I'm sure we can attain those statistics. The second would be to investigate biomarkers other than triglycerides associated with insulin resistance and the development of diabetes. And then the third would be to determine the link between the development of insulin resistance and the progression of cardiovascular disease. So our experimental procedures. Number one, the first procedure involves the utilization of alternative screening tests for insulin resistance, such as A1C, fasting plasma glucose, the oral glucose tolerance test, for comparison versus the triglyceride HDL-C ratio to assess the ability to accurately predict insulin resistance in patients. Procedure 2 involves the utilization of mammalian model organisms to induce insulin resistance within these organisms, and then evaluate them for biomarkers of insulin resistance through the extraction of blood samples and tissues for isolation and assay for analysis. Procedure 3 would involve the creation of a longitudinal study comprised of Black Americans presenting with insulin resistance to observe physiological changes associated with the progression from diabetes to cardiovascular disease. So we have some idea of some results that we may get from these procedures. The first would be that the easiest test to administer will be the A1C, and then second would be the fasting plasma glucose, and then third would be the oral glucose tolerance test, which that one is the hardest ones for patients to adhere to, because the fasting, not only the fasting requirement, but also how long it actually takes for patients to be inside a medical office to complete this exam it's hours long. So that might be a bit trying for some individuals to adhere to. So for the second procedure, so utilization of model organisms to search for other biomarkers of insulin resistance, it will present some challenges with translating the results to its relevance to human health, and that, well, fundamentally, there are differences, not only in obvious differences in our physiology, but there are also different pathways utilized by, let's say, rats in order to metabolize certain molecules in the body. And so we'd have to see how this would translate to human health. For the third procedure, the longitudinal studies can be pretty cost prohibitive, and sometimes you may lose participants along the way. There are, accidental deaths, or just people who drop out, who move, or are hard to follow up with. So that one may prove to be pretty challenging. So here's a bit of problem solving for those procedures. So for the first procedure, what we can do is we could utilize less invasive screening tests. So we could use, for instance, the waist circumference ratio, BMI measurements, and also blood pressure to assess the ability to predict insulin resistance in patients, instead of the oral glucose tolerance test, or even the fasting plasma glucose. So for procedure number 2, what we could do is, instead of utilizing mammalian model organisms, we could sort of create some cell cultures inside a laboratory and do some in vitro experimentation. So we could isolate different tissues and cells, human cells and tissues. So we could take liver cells, adipocytes, and muscle cells, and we could induce a type of insulin resistant state, and then analyze those cells and tissues for certain increases or changes in metabolites or molecules. For the third procedure, as an alternative to a longitudinal study, we could conduct an observational study, which would observe the same physiological changes, just over a shorter duration of time. Discussions and conclusions. So the triglyceride ratio will prove to be the less accurate at predicting insulin resistance in Black Americans when compared to other screening tests. This is something that I believe is sort of maybe controversial. There are some researchers saying that it does accurately predict insulin resistance in Black Americans, but there's obviously enough data refuting those claims. So the search for biomarkers other than triglycerides to predict insulin resistance. I believe this is a kind of thing that will involve collaboration with multiple departments and agencies to produce sufficient data. And third, longitudinal studies, which include data from diverse ethnic groups, will broaden our understanding of insulin resistance, diabetes, and cardiovascular disease across all populations. So for broader impacts, the reduction of the burden of health costs in the U.S., that will be a significant way to maybe provide not only more resources, but also to lessen the burden on hospitals and clinics for treating patients with insulin resistance and diabetes. The improvement of health outcomes across multiple populations The updating of medical guidelines to support preventative treatment for metabolic disease. Also, the creation of public initiatives and educational programs to increase awareness around insulin resistance, diabetes, and cardiovascular disease. And finally, the launch of an organization dedicated to promoting the inclusion of data from diverse ethnic and racial populations in medical research. For future research, I believe collaborative efforts will actually be key to expanding the knowledge on the triglyceride paradox in Black Americans. And so through the sharing of information, we could probably elucidate the findings on insulin resistance and diabetes further. Future studies should also utilize data from longitudinal studies, not just this one, but in the field that is researching insulin resistance and diabetes, to explore the relationship between insulin resistance, diabetes, and the progression to cardiovascular disease. These are my references. Thank you for your time.